Hi, everyone. Thank you for joining us and welcome to today's webinar focused on your frequently asked questions. My name is Kristen Ferrante and I will be your moderator for today's webinar. Presenting is Dr. Weston Umstead, Manager of Technology here at Chiral Technologies. After today's presentation, our technical support team will be available to help answer any questions you may have. Be sure to check out the handouts section for a copy of today's presentation along with our current product list. Now I would like to introduce to you Dr. Weston Umstead.
Thank you, Kristen, and good morning and good afternoon to all of you who've tuned in today. As you are very well aware, this is a webinar that is focused on you, the customer. The topic for today is going to be frequently asked questions that we come across, myself and other members of the technical team. Some topics that we felt were important to touch on today and kind of put them in film version so that you can go back and reference them in the future. There's three main ones that we're going to be touching on today, and they are listed here. The first one is, I have a chiral molecule. What column should I use to separate it? Second, my column isn't working properly anymore. What should I do? And then third, why isn't my chiral molecule separating? So let's dive in and begin to address this first question. I have a chiral molecule. What column should I use to separate it? Admittedly, this is not a very straightforward question to answer, and we need a little bit of background information before we can really explain why our suggestion is typically what it is. So to begin, let's look at our chiral selectors. As you are probably very well aware, we have many. If we're looking just specifically at the polysaccharide chiral stationary phases, we have 24 of them available. Chiral Pack IJ is now available in 3 and 5 micron. There are, of those 24, 18 unique selectors, as there are some that are shared between coated and immobilized versions. So 5 are available as coated and immobilized. And then IB is a little interesting. You have both IB and IBN, same selector. There were some chemistry tweaks that took place for the immobilization process to help it perform a little bit more like OD, which is the coated version. For your reference, Chiral Pack IA, shown here is the immobilized version of Chiral Pack AD. Chiral Pack IB and IBN is the immobilized version of OD. Chiral Pack IF, shown here, is the immobilized version of AZ. IH is the immobilized version of AS, and I mentioned IJ, which is our newest one, that is the immobilized version of OJ. You will notice in some cases, obviously a coated and an immobilized version, but you also sometimes have a choice between amylose or cellulose as the polysaccharide, as the case for Chiral Pack IA and Chiral Pack IBN. It's the same selector, 3,5-dimethyl phenylcarbamate, but IA or AD is on amylose and IB, IBN or OD is on cellulose, which might not seem like it's a huge deal, but it is when you begin to look at the helical structure of the polymer. This is not a polymer that just exists as a flat type of string-like material. It actually forms a helical structure. And so here shown is Chiral Cell OC. It's tris phenylcarbamate derivatized on cellulose. This, as it twists, forms the structure that you see here, and that structure, in doing so, forms these little pockets that we call chiral grooves. And you can see within these chiral grooves, the chiral selector forms all kinds of different planar configurations. And those configurations are going to dictate how and when the analyte traveling through the chiral stationary phase is going to react with or interact with the stationary phase. Now, within these chiral grooves, there are a number of intermolecular interactions that we can expect to take place. Some of those are listed here. For us with these polysaccharides, namely, we're looking at hydrogen bonding, steric hindrance, pi-pi interaction, dipole-dipole interactions. And what we need is at least 3 of these interactions to take place between the chiral stationary phase and the analyte in order for there to be some sort of discrimination between the pair of enantiomers. And this Easton-Stedman model demonstrates that fairly well. This is for a drug binding site, but you can imagine that rather than it being a drug binding site, this is the chiral stationary phase selector. And in our case, active enantiomer is going to be the more strongly retained enantiomer. The inactive enantiomer is going to be the one that is eluting first or faster. So as the enantiomer is traveling through the mobile phase and interacting with the stationary phase, there are at least hopefully 3 points of interaction where there's some energy difference that takes place. A, for instance, could be hydrogen bonding. B could be pi-pi stacking, and C could be some steric inclusion. So the active enantiomer comes through. The inactive enantiomer then has obviously the same interactions. However, it, because of geometry, doesn't line up as well. And so there will be an energy difference between these two that allows for some discrimination and therefore separation. Even if you were to take the active enantiomer and rotate it, it does not line up as well as the active enantiomer, and so you still don't have the same kind of energy that exists. In reality, the situation is even a little bit more complicated than that because everything is going to be solvated with mobile phase, and so this demonstrates that here where your chiral selector is solvated in certain spots by whatever the mobile phase components are, as well as your analytes. In the bound state, the ideal fit or the active enantiomer is likely going to have a fair amount of solvent exclusion, and the non-ideal fit or the inactive enantiomer, as it were, will still probably be solvated, and the solvation status has something to do with the type of separation that you see. So for instance, you might have a nice separation on hexane ethanol, but that won't exist if you go to hexane IPA, and that's likely explained because of the solvation status and interactions that exist but could be blocked or not as favorable as they were in another mobile phase combination. Well, if we go back and we look at Chiral Cell OC and we compare it to its amylose version, which would be Chiral Pack AC, I have that in quotations here because it's not an actual chiral stationary phase that is commercially available. But for demonstration purposes, you see that obviously both structures form these chiral grooves. They're a little bit more defined with this Chiral Cell OC than they are here with the Chiral Pack AC. They're certainly a little bit larger. The orientation of the selectors is obviously different. And so what we would expect to see is if we had a separation on OC, it would probably be very different on Chiral Pack AC. It could be different in a number of ways. One, it could not separate at all, or we might actually see some reversal of elution order, as is the case here for the separation of rulene. Up top here, rulene separates very well on Chiral Cell OD, which is the coated 3,5-dimethyl phenylcarbamate on cellulose. With heptane ethanol as a mobile phase here, nice 2-peak separation. We don't necessarily know which enantiomer is coming out first, but if we look at this chiral detector trace, you see here that the positive inflection is coming out first, the negative inflection is coming out second. Well, if we perform the same separation on Chiral Pack AD, which is again 3,5-dimethyl phenylcarbamate, but on amylose, same mobile phase, we see what could potentially be just a better separation, but we don't know anything again about elution order until we look at the chiral detector trace and we see that in this case, the negative inflection is actually coming out first, the positive inflection coming out second, which is indicating that we actually have a reversal of elution order. This doesn't always happen when we go from AD to OD or amylose to cellulose, but it can, and it can be explained by the differences in the geometry of these chiral grooves and the interactions and so forth. Another example here on immobilized columns, just to demonstrate that it's not just a coated phenomenon. This is a separation of levomethorphan and dextromethorphan on Chiral Pack IC, which is 3,5-dichloro cellulose. And if we look then at the 3,5-dichloro amylose analog, Chiral Pack IE, we do actually, again, see reversal evolution order. In the case of IC, dexmethorphan comes out second, and in the case of IE, dexmethorphan comes out first. So very interesting phenomenon that I said, again, can be explained by the shape and the size of the chiral grooves and so forth. Well, the mobile phase can also have a pretty large effect on the separation as well. As you can see here, this is a separation of another pesticide on Chiral Cell OC. Up top, you see that under 100% methanol conditions, the positive enantiomer comes out first, the negative enantiomer comes out second. As you begin adding in ethanol, we see the separation begin to collapse until we actually have complete coelution here with a 1:3 ratio of methanol ethanol. And then if we go 100% ethanol, it doesn't actually look like the positive enantiomer moves all that much, but the negative enantiomer pops out the front side. So solvation status has certainly a role to play in this separation. And so all of these examples are really to give you the answer to the question, which is what column should I use? You have to screen the compound. That is the answer. We really don't know how the compounds are going to interact with the chiral stationary phases under different mobile phase conditions. And the easiest way to find that answer is to screen it. So this is sort of a pseudo drawing of a separation of a screening setup where you can quickly go through 12 columns and a variety of different eluents overnight and come in the morning and find hopefully some baseline resolutions that you can begin optimizing for further work. This is an example of what that output would look like. This is a normal phase screening with 90-10 hexane ethanol for plus minus cannabicyclol. This is a very promising screening, I will say, in that you've got 5 results down here in green that are already pretty much baseline. In yellow here, you do have some partial separations if you were able to zoom in a little bit closer. And you have just 3 columns that came from the screening that only showed a single peak. But this is an example output of what that screening looks like. Really easy to set up overnight to get the results, as I said, to begin your optimization process.